All right, so why don't we get started? So good morning, good afternoon, evening, anybody wherever you're listening from. And I guess welcome back after a couple months off for summer. We have Ria, if you want to start sharing the slides. Thank you. All right, so we have a really nice symposium. Two great speakers lined up today. Professors Osvaldo Gutierrez and Rob Britton. You'll hear from them in a moment. But just a couple of quick announcements from Scott and myself before we get into the lectures. So I think a lot of people have gotten a lot of questions about the upcoming spring 2022 ACS meeting. So I'll say that I think everybody's aware that the fall meeting was a hybrid meeting. The Division of Organic Chemistry specifically, we did not have any in-person programming. We kind of felt at the time that that was the right decision based on the COVID situation. Going into the spring, we're cautiously looking forward to a return to at least some in-person programming. The COVID situation will obviously dictate how much is in person. But I think, as you can see from the slides, and this will be posted on the Division of Organic Chemistry website, we have quite a significant lineup of both award and organized symposia. I won't go through all of them, obviously, right now, so it take forever. But it should be a really nice meeting. And again, we hope that a good portion will be in person. Obviously, the COVID situation will dictate, but abstract submission for that conference, which allows you to choose what your preference is, whether it be virtual or in person, is already open and will remain open through October 11th. And I just also like to point out that we have a number of award symposia. The 2022 award nominations are going to open starting in fourth quarter for both the ACS National Awards and some of the Division of Organic Chemistry specific awards, like the Early Career, Mid-Career Investigator, Technical Achievements. We just yesterday announced our first cohort of industrial mid-career investigators, and they'll present, is actually the 2021, they'll present in San Diego, and then we'll have a second cohort at the Chicago 2022 fall meeting. So next slide, please. So I also wanted to point out, along the lines of cautiously returning to in-person programming, that the NOS, which was scheduled for 2021, has been rescheduled for next year, June 26th through 30th. And I'll point out that we are now accepting abstracts for posters. Registration will open soon. We have a really nice speaker lineup, and if you actually join the Division of Organic Chemistry, which costs $15 a year, you actually save $50 to $70 on registration. So just a little bit of a reminder to join the division. No, we don't have a slide for this, but I should also point out that the Graduate Research Symposium in New Mexico is going to be in person in November. So with that, I will turn it over to Scott, and also quickly congratulate Scott, who will be joining the Division of Organic Chemistry Executive Committee as virtual program chair, starting right now.
Thanks, Steve. It's my honor to be part of this, and thank you for having me as part of this program. It's been fantastic the last couple of years, and thank you everybody for joining each and every month that we present this symposium. First announcement today is a slide here from a Women in Chemistry Career Conversations virtual symposium featuring a number of speakers and panel discussions from ACS career advisors and Pfizer scientists about careers and opportunities for women in industry. So please sign up. It is a free event. The register link is there if you have a one of those QR readers. We also, you'll also be able to find this on the ACS website and other social media points such as LinkedIn and on Twitter. So definitely check this one out if you're interested in seeing what it's like to work in industry and meet some of these fantastic speakers and scientists. Next slide, please, Ria. All right, and next month we feature another DOC virtual symposium. The third Wednesday is October 20th, and we will be having talks from Martin Eastgate, a director of process chemistry from BMS Chemistry, and also from Sophie Rousseau, who is a professor at the University of Toronto. And so we're really excited to present those two talks to you folks next month, same place, same bat channel. Okay, next, and then in November, really super pleased to announce the first edition of what I hope will be a recurring theme for us is a PUI-focused virtual symposium featuring 3 talks from some outstanding professors at undergraduate institutions. We're going to have Rebecca Lalonde from Reed College, Miles Johnson from the University of Richmond, and Jeff Johnson from Hope College in Holland, Michigan. And so I really hope you guys all turn into that. This symposium is going to be really excellent to highlight some of the wonderful work that folks are doing at the Intro to Chemistry and combining teaching with research at these really vital institutions. So that's in November. Yep. And if you have any questions, comments, feel free to tweet us here at ACS Organic, or the 3 of us, Carl, who is not here today, unfortunately, and Steve and myself. We are all always on Twitter or this close to it, and so that we can respond or hear some feedback. So please feel free to reach out with comments and questions at any time. So without further ado, I'm going to introduce our first speaker, who, Osvaldo, if you want to go ahead and start sharing your slides, I'll do a quick intro. Our first speaker today is Osvaldo Gutierrez, and he is currently at Texas A&M University. He's originally from Salamanca, Guanajuato, Mexico, and he was raised in Sacramento, California. And while he was studying at UCLA, pursuing medicine, he actually worked with Professor Hauck as an undergraduate in chemistry, and inspired him to change his career path to organic chemistry. And I think we'll see the fruits of that today. After his graduation with a dual BS/MS degrees from UCLA, he moved back up to Northern California for graduate school at UC Davis. And there he worked with Professor Tantillo, completing his PhD on the use of computational electronic structure methods to reveal mechanisms of catalytic processes. A postdoc in the Kozlowski lab at University of Pennsylvania followed, again studying computational experimental tools to study transition metal catalyzed processes. In 2016, he joined the faculty of the University of Maryland as assistant professor, and subsequently was promoted to associate professor with tenure. And earlier this year, he moved to Texas A&M, where he is speaking to us from today. He is a recipient of several awards, including an NSF Career Award, and was recently named last year in 2020 as part of the CEN Talented 12. And along with his research interests, he's very active and passionate about increasing diversity in the sciences. And according to his website, it says his free time, he likes to listen to 90s rap and enjoy time with his daughter, 2 daughters and wife. And today his talk is combining theory and experiment to develop selective iron catalyzed multi-component radical cascades. And I'm really excited to hear all about it. So take it away, Osvaldo.
Thank you, Scott, for the very kind introduction. And it is really an honor to be here and participate in this virtual symposium. So with that, I really want to thank the Center for Selective CH Functionalization and the ACS for Division of Organic Chemistry. And today, what I want to tell you about it is a story that we've been developing in our lab since we started our exploration using computational and experimental methods. And that's really in iron catalyzed multi-component radical cascade reactions. And first is in understanding how the systems work, and then using that information to quickly design new transformations in a rational manner. So with that, first I want to acknowledge the graduate students and the people actually doing the work. So in particular, Wes Lee is currently at the postdoc at NIH. He's the one that really did a lot of the computational studies on this system. and more recently, Madeline Rotella, Cassandra Yushaw, Shuai Jin, and the postdoc that is really leading and spearheading a lot of this research is Dr. Lei Liu, and more recently, Dino and Angel as well. And I also want to acknowledge and take this opportunity to acknowledge the sources of funding, the NIH, the NSF, the Camille Dreyfus. And I want to take this moment to also promote the ATSI organization. So if you're interested in increasing diversity in the chemistry departments or any science department in your institution, please do feel to reach out. We're always looking for new opportunities to collaborate. So with that in mind, when I started my independent position in 2016, we went with the focus of studying open shell transformations. And with that, what we really mean to understanding radical processes, that being carbon-centered radicals and their reactivity, or metal-centered radicals and their reactivity. So our overview is shown here, where the aim goal, the overall goal that we're trying to achieve is to get things that you could easily make or buy, let's say for Sigma Aldrich, and using abundant and inexpensive transition metals, oftentimes non-toxic like iron and nickel, then we could quickly add value to these compounds. For instance, you could control the anti-selectivity in this process, or you could functionalize some of these bonds in these transformations. And hopefully that will translate to practical applications in agrochemicals, pharmaceuticals, materials, and so on. And in particular, the class that we're trying to focus on is this class that has really been evolving in the past 10 years in radical cross-coupling reactions, in which you have racemic, let's say, radical precursors. This could be potassium boronates, or it could be alkyl halides, redox active esters, and so on. And in the presence of light, a photocatalyst, a transition metal, you're able to generate alkyl radicals, and controlling the fate of this alkyl radical is key for the selectivity. One of the main transformations, you quickly trap this radical with a transition metal. And if you have the proper chiral ligand, you might be able to control the anti-selectivity. There's a lot of work to be done on that area. Another one is really to engage this radical with the solvent itself, generate a solvent addict, and then functionalize in a way that the solvent in the systems. We're interested in this two-component transformation and also in multi-components, so trapping that radical with some other reagent. For instance, a pi system in this case, in which you control the addition of the radical, addition to the pi system, and then trap that radical and potentially do an anti-selective transformations. So how are we able to control this radical? We really use a computational and experimental approach. So we do extensive transition state calculations on these processes to understand the factors that control the reactivity and selectivity. And then we go into the lab, or in parallel, we make different synthetic probes to test the mechanisms. And that generates fundamental knowledge. And from this, we hope to develop quickly new transformations. And I will tell you how we're able to achieve that. So the story that I want to tell you about today is the iron catalyzed cross-coupling reactions. Again, I mentioned that we work a lot with experimentalists in nickel, understanding nickel chemistry and photoredox chemistry. But the one that we do in our lab, both the computation and experiments in iron catalyzed cross-coupling reactions. So this is a very old field of chemistry and organic chemistry. So it's dated back into the 1950s, where you're able to engage a wide range of nucleophiles and electrophiles and couple those together in the presence of catalytic amounts of iron. And what I want to highlight in terms of the iron, not just the little men here, but also the LEGO systems, that these systems are in particular attractive because you're able to engage both sp3 hybridized electrophiles and nucleophiles. And that means that you could potentially not just make flat molecules that is typically done in pharmaceutical chemistry. There's many different practical methods, but also start tackling problems, for instance, in this drug where you have sp3 carbon bonds with new stereocenters. And that will be the overall, the long-term goal of our program, to quickly develop new methods to do these types of challenging transformations. And if we're able to achieve that, I think that's going to have broad implications in the synthesis of organic molecules. In particular, I think direct applications in medicinal chemistry, where already most of the carbon-carbon bond formations are done to transition metal cross-coupling reactions. And if we're able to at least provide or demonstrate that iron could do some of these reactions that are typically limited to palladium, then it could have effects in terms of the cost, abundance, and toxicity. And hopefully that will translate to cheaper medicines for everybody. So when I started my independent career, I started thinking, which of the many different reactions that we could start analyzing using computational tools do we want to focus on? So I spent a lot of time thinking about this problem, and Fortunately, I ran into this very recent paper at that time, basically a year before I started my independent position, where Masahiro Nakamura from Kyoto University published this report that was really transformative in a way that it turned out to be the first in anti-selective iron catalyzed cross-coupling reaction. And in this one, they used alpha chloroesters in the presence of catalytic amounts of iron and a very common chiral ligand in granular. You're able to basically do alpha aerylation, so carbon-carbon bond formation with good yields and modest anti-selectivity. So here, in terms of the mechanism, it was kind of clear or suggestive that you might have radicals involved. For instance, presumably you cleave this alkyl halide bond, you generate a radical, and this radical you could trap with some iron, presumably with a chiral ligand involved, because that will lead you to an anti-selective transformation. Or this radical could cyclize and do 5x cyclization, and then you could trap this radical with another species, but it was not clear why this was not, you're not able to control the stereocenters. Even in the presence of a new group here that will create a new stereocenter, you're not able to control anti-selectivity. So for us, this was crucial. If we're able to understand the mechanism of this transformation, I think that will provide a lot of information for the field of iron catalyzed cross-coupling reactions. In addition, if we're able to not just understand this reaction, but also start developing these radical cascades and seeing how we can manipulate this substrate in order to promote any anti-selective transformations, I think that will be even more transformative, because we can show as a proof of principle that we can do mechanistic studies and follow up by doing experiments to develop new reactions in that sense. So I won't take you through all the computations that we did on this work. You can read this paper here that we published in 2017, in which we were able to, I guess from the computations, they show that potentially an iron 1 species, a very controversial iron 1 species, with one ligand in the quartet spin state is responsible for activating the alkyl halide to generate the radical in an iron 2. And controlling how those two combine together is crucial for selectivity and also in anti-selectivity and reactivity. What we see is that, consistent with experiment, if you had the granular basically in one pot or fast, what presumably could happen is that this iron could do quickly transmetallation before the radical rebounds to the iron, and that will lead to unselected biaero formation. Computations show that all pathways from the biaero species always forms underside products. However, if you're able to slowly add the granular, then you allow time for the radical to rebound with iron 2 and enter selectively, generate iron 3, and finally reductive elimination will give you the product, iron 1, followed by a transmetallation step, and you continue the catalytic cycle. But what's also clear from this analysis of this diastereomeric transition states that, consistent with experiment, what we see is that the major enantiomer, it can be rationalized based on this stereochemical model, which we have this, presumably this donor-receptor interactions that I'm going to call. And if that's true, then it should be, I guess, observable in experiment that if you change the properties of this arrow group, that should translate to changes in selectivity. And that's exactly what was observed by Nakamura, even though it was no explanation as to why you saw those changes in selectivity as you vary the electronic properties of the arrow ring. Also consistent with experiment was another observation by Nakamura that you really needed to have bulky groups, and in this case, a thepto group, in order to induce high levels of selectivity. So as you go from methyl, ethyl, isopropyl, and terbutyl, and so on, you start increasing selectivity. So that becomes at least consistent with this other diastereomeric transition state, the one that you don't want, that will lead to the opposite enantiomer. Because you start, as you bulk it, this group, you start destabilizing these interactions, and that will raise the energy, and you will have a more selective transformation. So with this in mind, we're very fortunate that at the same time, Morikuma and Nakamura collaborated, and they published basically what I think is, given the complexity of this system, almost identical conclusions, in which we agree on the radical addition to the iron 2 as the key step to form an iron 3 finally by reductive elimination. And we disagree in the order of the transmetallation. So to differentiate between the order of the transmetallation, we really need to do more experiments. And in particular, we're collaborating with Mike Neidig at Rochester to elucidate and hopefully get more insights into this transformation. If we're able to do that, then that will provide even more information, fundamental information about this mechanism of this transformation. And then my goal is to really use that information to develop quickly new asymmetric transformations. So look for that in the future of where our program is going. So now we can go back to this example that before it was challenging to understand as to why that is the case. So here, Nakamura observed that you're able to basically activate this carbon halide bond and do aerylation at that site and then selectively. But if you allow for the radical cascade, we could still trap the radical, presumably the radical, to generate carbon-carbon bond, but that was racemic. So now we can understand this result based on our very simplistic stereochemical models. This group here basically lacks the ester group that is critical to stabilize that desired diastereomeric transition state. And it lacks this other ester group that is also critical to destabilize the undesired diastereomeric transition state. So now, if that's the case, then can we make a radical cascade and anti-selective? And the answer is yes. So we decided to build this substrate here to probe the hypothesis, presumably under identical conditions to generate a radical. You could trap this radical, and that's actually the major product, but we don't want that. We want to focus on radical cyclization to generate the cyclic radical or the cascade radical. And then this, in the presence of the chiral species, it should undergo an anti-selective transformation if in fact those two interactions are playing a role. And in fact, it does. Low in anti-selectivities, low yields, but nonetheless, it kind of shows that you're able to generate this radical cascades, this radical, and do some anti-selective transformations. However, it was not clear if this really was going through a radical process, or perhaps iron chemistry is known to do single electron transfer with other species, and presumably this could undergo a reduction to generate the enolate, and that could do other type of chemistry. So to differentiate between radical chemistry or enolate type chemistry, we decided to build a very similar substrate as the one shown on the very top. But instead of this alkene, we decided to focus on the vinyl cyclopropane as a radical probe. So we built the substrate, we subject it to identical conditions. This radical presumably undergoes a radical cascade. And now at this stage, if it's really going through a radical, it should quickly ring open. Those rates are well known. Generate a primary radical, then we can trap that radical. And we're able to achieve that. So we get primarily the radical cascade product in this transformation. What was really surprising from this is really the high yields that we were achieving. So we duplicated this reaction. And again, we were very skeptical about this result, but we keep getting high yields in this transformation. And as you can see from the ones in the top, typically the radical cascade products are really low yielding. So there are several different examples. Some of the best results are really around 20 yields or 20% yields for us. So in this case, we're getting high yields, 90% yields. So we decided, what if we combine these two concepts to develop an anti-selective radical cascade? So we decided to build a little more complexity to this substrate. So hopefully this is not the first thing that you would think if you were trying to develop a radical cascade process, but hopefully I can also convince you that now it becomes logical as to why we decided to build this particular substrate. We hypothesize you generate the radical as before, undergo a radical cyclization, as we shown before, and now this one in the presence of an ester group, now that could be involved in controlling that anti-selectivity, and you should be able to control the carbon-carbon bond formation at that site. In a practical sense, this is not something that you want to isolate or characterize because you have many different stereocenters. So we decided to oxidatively cleave and just to focus on these key steps. Can we control the anti-selectivity at that step? And the answer was yes. We're able to achieve of the products 80% yield, good in anti-selectivity, 8218. And we also got the other corresponding product, racemic, which is consistent, the radical cascade proceeding without the involvement, at least not in the close proximity of the chiral iron species. So with that in mind, we quickly decided to analyze the substrate scope. But in terms of the substrate scope, we didn't want to build many of these derivatives. That would take a long time. It took a long time to build one. So we instead decided to only focus on the granular scope, just to make sure that this is not just one example and it doesn't work for anything. And it was very pleasing to see that you have a wide range of nucleophiles you could easily make or buy. And it gives you on average good yields and on average 85-15 in anti-selectivity. For some cases, for this sterically hindered system, you get up to 90-10. And I would note that this systems here, these granules were previously low yielding from the other reaction. So in this case, you're able to engage other granules in this radical cascade. So we think that the radical cascade is behaving slightly different than the direct carbon-carbon bond formation. So with this in mind, we also acknowledge that I hope that no synthetic organic chemist will use this substrate to make something that looks simple as this and anti-selectively. There's many other methods. So we realized that if we really wanted as synthetic chemists to use our method, we needed to use something more practical. So what if you just simply cleave this bond, one of these bonds, and instead generate things that you could easily buy or make in a 3-component form, not a 2-component form? And we exactly did that. So we have the alkyl halide, we have the vinyl cyclopropane, and we have the Grignard. And in our first try, we got high yields with the ligand that was previously used for the 2-component. However, this is a very expensive ligand, and we're not making new stereocenters. So we decided to quickly screen things that we had in the lab or we could quickly buy. We screened some monophosphine, diphosphine, diamine ligands, and we're very fortunate to get our hands on ligand L11 that turned out to be excellent for this 3-component reactions, and actually very general for this 3-component reactions. We think now we understand why this ligand is special in comparison to the other ones where you make the cyclohexyl groups less sterically hindered. The idea is that iron is well known that it can coordinate 1 phosphine ligand or 2 phosphine ligands. And in this case, you can have this kind of equilibrium. If you want to favor this kind of species, then make this less sterically hindered. If you want to favor this kind of species, start bulking up. But if you overbulk this group, basically use very big groups, then you might have problems that the ligand will completely dissociate from the iron and not have a catalytic active species. So we think that our catalytic active species is something that looks like this, at least a mono coordinated iron species with an error group. And I'll show you some results where we're going with this type of chemistry. So with that in mind, we went and explored the nucleophile scope. Here's some examples on this, and I won't spend a lot of time. And also the electrophile scope, in which previously was all limited to alpha chloro and bromo esters. Now we're able to expand beyond alpha chloro esters and bromo esters to difluoroesters, or simply fluororadicals or tertiary radicals that could also play a role in basically work in this transformation. In addition, we're also able to mix basically derivatives or biderivatives where you change this group here or decorate the cyclopropane group. You can incorporate heteroatoms, and now you can even establish new stereocenters. And of course, this one here, the one with an ester, this is the one that we predict with the same identical chiral ligand that we used before, that should engage this non-covalent interaction that I discussed at the beginning from the computational studies. And in fact, that was the case. We get low yields for some, high yields for other ones. These are not optimized. So this is something that we could always go back and revisit. And we get good anti-selectivities for all those systems. Again, there's an auto-optimized, we haven't screened other ligands, but presumably maybe if we use virtuous screening using computational tools, we might be able to modify that ligand to make this even more anti-selective. And that's something that is ongoing in our lab. So with that, we decided to test another hypothesis. Is this reaction really limited to highly strained vinyl cyclopropane rings or highly reactive vinyl cyclopropane rings? Or can we use other physical organic concepts to promote this transformation? So if we go back to what we think is the mechanism of this transformation, you have an iron 1 species that activates the alkyl halide to generate this critical radical cage pair. And again, if you want to promote a 2-component reaction, what you want is this radical to engage with the iron, and you could do that. We've shown some examples to do that. But in this case, we want that radical to react with the alkene. So how do you go about doing that? And one thing that you could do is simply raise the barrier for this radical rebound. One straightforward way is to simply make this sterically hindered. So for instance, a tertiary radical. And if you really want to push the equilibrium to this direction, how about just make this alkene your solvent? And you will increase the chances of the radical engaging with the alkene to do regioselective addition to generate a secondary radical. Now the secondary radical is less sterically hindered, and that could rebound with the iron and do the type of chemistry that you want. So leading to one dicarbo functionalization of the alkenes. And I will note that this is unactivated alkenes without directing groups, which are still very challenging to do. So we did that, and we're very fortunate that actually with slightly modifications to the reaction parameters underneath, basically using the alkene as your solvent, we're able to achieve high yields for these transformations. I also will note that this also worked with both the alkyl bromas and alkyl iodines almost equally the same, basically similar efficiency for some systems. So in this case, you get good yields for using electron-rich or electron-poor Grignards. Also, you can engage vinyl Grignards, and dye-substituted things that you have aryl halides that you can do other further chemistry with this ones here. In terms of the alkyl halide scope, again, as shown, consistent with the mechanism that we're proposing, sterically hindered radicals, all tertiary radicals work in these systems. Here's some represented examples. And also fluoroalkyl radicals, which they go through a different type of mechanism. The fluoroalkyl radicals, they have a propensity, orders of magnitude much faster to engage with the alkenes. So in doing G's addition, so that you don't need high concentrations of the alkene to promote that type of transformation. So it allows us to explore more chemistry in terms of making fluorous compounds. And we're going to see how we're able to take advantage of this other type of reactivity or the propensity for radicals to engage with pi systems to do new types of reactions. What was kind of surprising on this end was really the scope in terms of the alkene. We were very surprised to find that this actually turns out to be very broad in terms of always adding to the less substituted alkene. We're able to do this in gram scale. We're able to also incorporate some key functional groups that organic chemists might be interested on, for instance, protected aldehydes, alcohols, ethers, esters, even in heteroatoms. And we're also even able to tackle basically internal alkenes. Again, this is limited to using fluororadicals because they proceed through, again, the kinetics are much faster to add to the alkene. So that allows us to make basically dicarbo functionalization of internal alkenes. So with this in mind, now I want to basically finalize this talk and spend more time on this work that is has been accepted for publication, but it's not out there. So hopefully you'll see the whole story soon. And we went back and started thinking, so what is known about iron chemistry? So there's a lot of two-component reactions about iron chemistry. And what is the type of mechanism that had been proposed in this transformation? And there's many different mechanisms proposed, but in general, a lot of them proceed through radical intermediates. And this type of radical intermediates in which you have different carbon bonds in the vicinity. So carbon-centered radicals with carbons in the vicinity, or alkoxy in the vicinity, or esters in the vicinity, like the one that I showed at the beginning of this talk. And you're able to trap these radicals with several different nucleophiles and iron carbon species with different sp3, sp2, and so on and so forth, changing the properties of the ligand and the scaffold. More recently, we got inspired by the work of Fu and Molander. We collaborate a lot with Molander in nickel chemistry, nickel photoredox redox chemistry. And they developed different methods in which they were to use either alpha haloboronates or binoboronates, sometimes even in 3-component reactions, where you generate alpha boron radicals, and they're able to trap this alpha boron radicals with a wide range of different systems. So here using nickel is well established now. But with iron, we thought, can this be possible? Can we stand the scope of this transformation to start trapping new types of radical, in this case, alpha boron radicals? If so, that could open up basically another area of chemistry that we could start capitalizing on, making this highly versatile boron reagents that could be used for medicinal purposes and agrochemicals and so on. So here's our strategy. This is what we're proposing. We're proposing that we're going to use alkyl halides, same thing as before. We're going to stick to grainers because we like grainers. In our lab, we have a lot of grainers, and we know that we can control some of the reactivity with grainers by simply adding it slowly. And we're going to test out if we're able to control basically selective addition to the binoboronates, generate the binoboronates, and then trap those binoboronates. So with that in mind, the answer is yes. By making slight modifications, keeping the same ligand scaffold that we show, again, this turns out to be very general. I will note that all these reactions really proceed at low temperatures and really fast. So we stop the reaction at 1 hour, but a lot of these systems, you could actually stop the reaction even at less time, 30 minutes or so. And the idea is that these ones work with a wide range of different nucleophiles, and you're able to generate the dicarbo functionalized products as you want with electron deficient, electron rich, monosubstituted grainers, disubstituted grainers, and so on and so forth. Even the heteroatoms will work. And even if this binonucleophiles, you can also trap it with a series of binonucleophiles. We even have one example where we're able to use basically an alkynyl grainer, and that gives you okay yields, poor yields in this case, but this is not optimized. This is using the conditions that were optimized for the aero grainer. So we think that we're able to optimize this method and also do further chemistry with this. So that's ongoing in our lab right now. In terms of the alkyl halide scope, again, we show that we can engage many different tertiary radicals, and here we have some representative examples. But also, we're also able to engage now secondary alkyl radicals. Again, now you have a more reactive, more electron deficient pi system. So now we're able to engage secondary radicals or tertiary radicals with ester groups. And now here you could imagine several different manipulations that you could do at the alkyl boron bond or the ester bond and do further chemistry. So is this just a method that is able to do the same things that nickel is able to do? And the answer is no. It could also do things that nickel is not able to do or it still struggles to do. And one of the things that it's able to do is that now we can engage a wide range of fluoro alkyl radicals. So here are some representative examples, some with really high yields on these systems. And we think this can provide direct access to alkyl boron reagents with fluorine attached to it. And this can be impactful for agrochemicals, like I said, materials chemistry and so on. And now we started thinking, can we develop more elaborate radical cascades? So I got inspired by a lot of the mechanisms that we go in Dean Tantillo's group during the Monday nights group meetings. And we always talk about radical mechanisms, and I got inspired into trying to develop new transformations, especially more elaborate transformations using radical cascades. So one thing that we saw is that we could potentially use this reagent. It's a commercially available. It could be a new radical precursor, and it could be useful to generate these types of products where it could be a 5-member tetrafluorocontaining carbocycles. And those, if we look at the literature, actually they're very challenging to make. So there's no general methods to make these compounds. So we started thinking, if we're able to use this in this type of chemistry, we do halogen atom abstraction to generate the radical cage pair. This fluorine radical, again, we don't have a problem with this radical rebounding with the iron because this radical tends to engage with pi systems in orders of magnitude much faster. So we will engage with the alkene, generate this type of radical. And now this radical has 2 fates. One, the one that we already saw before the reactivity, it could do carbon-carbon bond formation at this to generate that dicarbo functionalized product. Another one, it could do radical cyclization. And then you could promote a primary radical that less sterically hindered, and you could trap that radical and generate the desired transformation. So how do we go about designing this system? So first, do we even generate this product? And the answer is yes, we do have that product with identical conditions. And do we see any of this product? And the answer is yes, we do see some of that product, basically almost a 1-to-1. So how do we go about making this reaction more selective? So one of the typical things is simply shut down one pathway, shut down this pathway. The way we're able to shut down this pathway is simply by lowering the catalyst concentration. Now you allow more time basically for the radical to react with the alkene, 5x cyclization, and then you can trap that radical, and then now you're able to promote the transformation. So that for this particular reaction is 50% yield, and you have almost no dicarbo functionalized product. So we went on with that and started exploring the alkene scope. And we're very fortunate that many different alkenes work. So almost every single alkene that we got our hands on working for this particular system. So you can engage, of course, electron deficient alkenes, but also electron rich alkenes. For instance, this one shown here, even high yields, you have this vinyl ethers. Here's a crystal structure of the desired tetrafluorocontaining carbocycle. You could engage vinyl silane, vinyl amines, some with high levels of diastereoselectivity selectivity. And you could even engage exocyclic alkenes to give you the corresponding spirocyclic compounds, whether you have a 4-member or 5-member or 6-member ring. So here's one particular structure in which you maintain the stereochemistry at this site. In addition, again, internal alkenes also work to generate this fused product with tetrafluoro. So now if you look at the literature, start to think about how would you make something like this. Potentially, this could be a group that could be found in many different medicines. It's actually very challenging to make. So here it provides in one step a very straightforward access to this compounds. Now you can think about functionalizing or making derivatives or doing other type of chemistry. And also, yes, we just tested some of the functional group tolerance. We're able to see that it engages basically protected amines, some alkyl halides, and alkyl boron reagents so that you can do further chemistry, even alcohols under a slight excess of the grain. So in terms of the nucleophile scope, again, it shows the same thing as before. It engages a wide range of nucleophiles and granules. And we're even able to apply it to different alkenes that have more elaborate or more complex structures. For instance, this one shown here to generate the corresponding 5-member ring products. In terms of the radical cascades, you don't need this fluoride groups. You could also use tertiary, basically alkyl halides with an appendant allyl group, and those also engage and participate in this transformation. And again, in this case, you have to play around with the conditions to promote the radical cascade, but nonetheless, you can engage this one's in good yields. So we started thinking and going back to the things that we're really passionate about. What is the mechanism of this transformation? Or what are some of the insights that we could get from this mechanism? And one thing we realize is that I think everybody's well aware of this, that in terms of iron chemistry and start analyzing things with iron, there's no one general method to study these mechanisms. So what we really feed on or rely on is heavily mechanistic studies. And for that, we went with probably the world's expert on iron chemistry in terms of the mechanisms, Mike Neidig at Rochester. We collaborated with Camila Aguilera Cuenca, that is shown here, to get an understanding of what is the nature of the active species or the species involving catalysis. So as shown here, again, we think that this ligand is very special for iron chemistry. And one thing that we could quickly notice is that they're able to isolate many species that are highly reactive. They took a long time to characterize this species and to get this species. And one thing that is consistent, and no matter how much catalyst loading ligand loading you add, it is always consistent with only one phosphine ligand added to this systems. So you have an iron 2 with one phosphine iron monoaero species, this aero species, and the one that goes reductive elimination, the iron 0. It provides a structure information for us, and that helps us calibrate our methods. It provides also knowledge about the spin states of this system. So this one is intermediate spin, high spin, and high spin. So now, if we use just this knowledge, we could calibrate our computational method to which one is able to replicate the structure parameters and also the spin parameters. And now they even went further. They want to know that kinetics. So now we're able to calibrate with the kinetics as well. So which one is able to replicate all those three. And that helped us out basically to choose the method and do further exploration. And now from here, what we've seen from this very simple experiment, that you have a 0 equivalence, you have primarily this component, this orange species. After 1 minute and you start adding the granular, do transmetallation, so you get the mono arrow. As you add more granular, you start getting more of the this arrow, but you still have the mono arrow species. And then finally, you start getting the reduced species after 5 minutes. So these are very fast transmetallations. So which of those species is the one that's responsible for activating the alkyl halide? So one thing that was clear between the mono arrow and the bis arrow, the one that is much faster activating the alkyl halide is this bis arrow species. As shown here, you basically have unreacted mono arrow species, and the other one disappears, and now you have this one here. You have the green one here. You have this it's iron 2 species that then, if you add more granular, you could convert it into the mono arrow species and so on and so forth. So then we did some more mechanistic studies. So we decided to explore the chemistry with this vinyl cyclopropane, again, consistent with a radical process. You do form this key species, this radical that can ring open, that's predicted to be much faster, and then trap the primary radical. So here's the one that we observed. Again, you could also engage with E or Z alkenes, and it gives you the same diastereomeric ratio. That would be consistent with radical addition, followed by equilibration from sigma bond rotation. So then this computation, where again, we use a lot of the knowledge from Mike Neidig, the structural parameter, the spin parameter, to guide these results. And what we see is that once you generate the radical, presumably through the bis arrow species, iron 2, you do radical addition, low barrier, to generate this alpha boron radical. And now you could quickly trap that alpha boron radical to generate an iron 3 and then reductive elimination. If you see this goes through an inner sphere mechanism, so that means that this radical is in close proximity to the iron species, to the ligand species. So that gives us hope that we might be able to rationally in silico start predicting modifications about the ligand to promote enantioselective selective transformations. And we have very exciting results on that area that we could talk offline about those. And now here we can also show that the one that outer sphere is actually much higher in energy. So we think that the mechanism is going through inner sphere carbon-carbon bond formation with this monoaero species. So here's the overall catalytic cycle that we think. We have evidence for many of this from the grains from experiments and also a mix of organic synthesis. And the other ones where it's more challenging experimentally to get ideas about the transition state, we use computations, again, heavily benchmark against structure and electronic parameters and kinetic parameters obtained by the Neidig group. And finally, I just want to thank everybody for listening to my talk, and hopefully I can convince you that using this approach of organic synthesis and computations in the same lab, will quickly lead to developing new reactions and new mechanistic understanding. So here we went from try to understand a very simple two-component reaction to using this knowledge to develop an anti-selective radical cascades, three-component cascades, and now other things that I think they're going to be valuable for synthetic community. For in particular, the making this tetrafluoral containing carbocycles, I think that provide the direct route and hopefully will translate to practical applications. So with that, I'll be happy to take any questions. And again, thank you all for being here.
All right, thank you, Osvaldo. This is wonderful talk. So much chemistry in there. That's awesome. If anybody has questions, please enter them into the Q&A in the chat, either in the Zoom or into the YouTube channel. We'll be monitoring both. So I have a question first off back on the, of course, on the tetrafluoro compounds that you were talking about. You have two, you're bringing in two olefins with it's just amazing the selectivity that they don't have any cross reactivity, you know, whether you insert into one, but it just seems like you get maybe some dimers there of the difluoroolefin reacting as well. You don't see that in your product.
We don't see it with this particular system where we do this radical cascade. We do see it somewhat with the dicarbofunctionalization product. We do see some of that, but not, you can control that. And we think that the reason why we don't see much of that is it really comes down to once you form this radical, then you have a choice of this radical adding to the iron species that you have in solution, or this radical adding to another vinyl boronate. In terms of the concentrations, I think at this stage, basically the barrier for this one to add is almost barrierless to add to the iron. So it will quickly be trapped before it actually escapes this, I guess, solvent cage or where this one is, and do other chemistry. So the barrier for radical addition to another alkene, and that's the limiting reagent, is much higher. I think if you increase significantly the concentration of the vinyl boronate, then that will lead to problems there. And in this case, it's slightly different because you have basically this radical cascade. So now you have, once you add one, can this radical engage with another alkene, and that becomes less likely on that sense. So we're really, I guess the one thing that I want to mention is that the grainers, by adding it slowly, we're also able to basically limit the concentration of the grainer in solution, because we're slowly feeding the grainers, not all one pot, and then that avoids some side products that you might form on typical one pot procedures.
Gotcha. And is this something you can do with could you do this with zincates as well? Would they do a similar, instead of the Grignards?
We only did one example, and so far that has failed. So the grainer is behaving significantly different than the zinc reagents. But like I said, we only tried one just to see if we get somewhat a similar reactivity, and so far it's not. But we need to explore those aspects, but because there's a lot of attractive features about using zinc reagents rather than grainer reagents.
That's excellent products. You're just really diverse products that you're creating through this chemistry. It's really, you know, from really simple starting materials into some very wonderfully complex architectures. So at this, I'm not seeing more questions now on our other chats. I think anybody has any more questions, we have definitely a bit of time here for some questions. Steve, are you thinking of anything here?
Well, I mean, I guess, you know, based on the time, why don't we move on to Rob's talk? But if people have questions, we can, you know, you can still use the Q&A function or chat box. And I think, you know, both speakers will remain for a few minutes after the talk to address them.
Yes, I do see that. Yeah, we are right at the switch over time. So thank you. Thanks for noticing that. All right, Osvaldo, thank you very much. Wonderful talk. Thank you for presenting, and we're really glad to have you here today.
Thank you. Appreciate it.
Yeah, really nice talk. Yeah, great chemistry. All right, so I'm very excited to introduce our next speaker because I've had the pleasure of collaborating with him over the last couple of years. So Professor Rob Britton from Fraser University. Rob grew up in Ontario and completed his bachelor's at the University of Waterloo with Victor Snikas. And at the same time did co-ops at Bayer and Aptech. He then moved to University of British Columbia, where he earned his PhD with Professors Raymond Anderson and Edward Piers in natural product isolation, structure determination, and total synthesis. He completed an NSERC postdoctoral fellowship under Ian Patterson at Cambridge in the total synthesis of complex macrolides. And then after a couple of years at Merck Frost, Rob joined the faculty at Fraser in 2005, being promoted to associate professor in 2011 and full professor in 2015. In 15 years, Rob has authored around 75 publications with a focus on bioactive, densely functionalized natural products and biomolecules, and then you'll see some of that today. For his extensive contributions, Rob has earned a pretty long list of awards, just a few of which are the Bioorganic and Medicinal Chemistry Award, the Innovation to Commercialization Award, and the TIMI Chemistry Journal Award. So today he'll talk about a short de novo synthesis of nucleoside analogs.
Thanks for the introduction, Steve. And I want to thank Scott, Steve, and Carl for the opportunity to speak today. So just by way of introduction, so my group at Fraser works in several different areas. So in one part, we, sorry, I'm just going to get this laser pointer going. Here we go. So in one part, we work on development of methods, primarily methods to support drug discovery. So late stage fluorination or radiofluorination and the synthesis of stereochemically rich heterocycles. We're also quite interested in natural products, and particularly stereochemically rich natural products that have potentially useful biological activity. These method development efforts also support nuclear medicinal chemistry efforts in our lab. And maybe a little bit more uniquely, we also work on the structure determination and large-scale synthesis of insect sex pheromones for the purpose of development of environmentally friendly traps for insects. So what I want to talk about today, though, is our work with Merck, actually, on the synthesis of nucleoside analogs. And our interest in nucleoside analogs comes from the fact that these are really the largest class of small molecule antivirals and really key molecules in oncology. In general, these molecules mimic endogenous nucleosides or nucleotides, and they can act as antimetabolites and often inhibit RNA or DNA polymerases, nucleoside phosphorylases, and other important enzymes especially in viruses. With regards to structure, I've just shown 3 examples here, and these are often modified at the so-called C2 prime position, as in the nucleoside core of sivosbuvir, the C4 prime position in this molecule 8K or MK8591, or they have unnatural nucleobases in the same molecule. You can see this in MK8591 or in ribavirin, which is a broad spectrum antiviral. Now, obviously, in the current pandemic, these small molecule nucleoside analogs have been very important, and remdesivir was the first therapy approved by the FDA for COVID-19, and molnupiravir is another molecule that's of interest right now and entering phase 3 clinical trials. So despite the I would say apparent simplicity of these molecules, they do present challenges for synthesis, both to medicinal chemists and process chemists. And just a few years ago, the synthesis of non-canonical nucleosides was highlighted as an emerging area of high potential impact in an article in Science. And just to highlight some of these challenges, oftentimes when people tackle the synthesis of a nucleoside analog, they start with a chiral pool starting material. And this limits the structural diversity and really the stereochemical diversity that you can access in the ribose core of the molecule. Another challenge is if you want to modify your C2 prime position, you need to protect C3 prime and C5 prime, and this can be difficult, especially on scale. And so there's been a lot of great work devoted to trying to find ways to selectively protect these functional or these alcohols. Modifications to C4 prime, such as in this molecule in the top middle here, often require really lengthy syntheses that involve protecting group manipulations and redox chemistry. And the addition of heterocycles to the C1 prime position to attach the nucleobase, especially when the sugar core is modified, can often lead to mixtures of anomers. So there's lots of area here to improve upon. And so clearly, there's been great effort in the de novo synthesis of nucleoside analogs. And I'll just highlight a few examples. This is an example from Macmillan's group a few years ago where they showed a Mukayama aldol reaction using this alpha chiral aldehyde, gave this stereochemically rich building block that we converted into the core of sivosbuvir in just a few steps. And just last year, a group from Merck Process showed that the addition of these really unusual trimethyl aluminates or acetylenic trimethyl aluminates to this chiral ketone could provide a key stereocenter, what would become the C4 prime stereocenter in MK8591. And so my group at Simon Fraser has spent, I'd say, many years now exploring the use of alpha chloraldehydes as building blocks for these stereochemically rich heterocycles. And this work was largely inspired by two independent reports in 2004 by Jorgensen and Macmillan on this enamine catalysis, or using chiral amines to catalyze the asymmetric alpha chlorination of aldehydes. And more recently, Matthias Christmann has made some really important contributions in this area as well. Now, one thing we found over the years is that while we can make these molecules and use them as linchpins in heterocycle synthesis, they're quite unstable, they're difficult to store, they can epimerize and degrade easily. And so a few years ago, we came up with an alternative procedure for making and exploiting alpha chloraldehydes, which I'll just show you very quickly here. So in this process, we take proline as the catalyst and N-chlorosuccinimide as the chlorinating agent. We generate a racemic mixture of alpha chloraldehydes via the chlorination with proline. And in fact, proline catalyzes the racemization of this alpha chloro stereocenter. But that doesn't matter because in the same pot, we couple in an aldol reaction. So in this case, with this protected dihydroxyacetone derivative or dioxanone, and this can give us either an anti-aldol product with a syn-chlorohydrin or an anti-aldol product with an anti-chlorohydrin. And in fact, this second step, which happens in the same pot, affects a dynamic kinetic resolution. And so in this one pot process, we convert these readily available aldehydes then into these stereochemically rich chlorohydrins that we've exploited in several synthesis campaigns. And just to demonstrate the kind of things that we do with these, you can carry out a carbonyl reduction or reductive amination, followed by a cyclization, and you get quick access to these either polyhydroxypyrrolidines or ribose type analogs. And so it seemed logical that this chemistry would apply itself to nucleoside analogs. And I'll just highlight that we have exploited this chemistry in both the total synthesis of natural products, the synthesis of natural product-like molecules. This chemistry has allowed us to access a variety of mechanistic probes for glycoside hydrolases and also support some medicinal chemistry efforts in my lab. So this was the idea then. We would exploit this alpha chlorination aldol reaction, this one pot process to make these types of building blocks, reduce the carbonyl and cyclize. But this turned out to be a really, I'd say, a horrible idea. These alpha heteroaryl aldehydes are just far too unstable to use even transiently and capture in the same reaction. And so this idea kind of got shelved for a few years until a really fantastic graduate student, Mike Meanwell, joined my lab. And he had the idea to, instead of doing alpha chlorination aldol, an alpha fluorination aldol reaction. Now, there's obviously good precedent for the alpha fluorination of aldehydes, and several groups published in the same year, back in 2005, the fluorination of aliphatic aldehydes using NFSI or select fluor as the F+ source. The problem with these molecules, though, is that they're even less stable, I'd say, than the alpha chloraldehydes. These polymerize, decompose, and epimerize quite easily. And so generally, they're only isolated after reduction to the corresponding fluorohydrin. So we would have to get lucky here. And the real question then was, would these alpha heteroaryl aldehydes be stable enough to capture in this one pot reaction? So the second aspect of this sequence would be to reduce the carbonyl and affect a cyclization or displacement of the fluoride. And we had pretty good confidence that we could carry this out. And there's a really fantastic paper by Carmen Ortiz-Millet back in 2016, where they made this fluorinated alkyl carbamate and showed that you could displace the fluorine using either oxygen nucleophiles or carbon nucleophiles And in the case of oxygen nucleophiles, this occurred with retention of stereochemistry, and with carbon nucleophiles, they recovered mixtures of alpha and beta anomers in the product. So it showed this could go via an SN1 type process. So this work was started, as I already mentioned, by Mike Meanwell, who was a graduate student in my lab at the time, and he was assisted by these postdoctoral fellows over the course of a couple of years. But really, this work benefited from a fantastic collaboration with some scientists at Merck. Steve Silverman, who obviously introduced me here today, L.C. Campo, and Ryan Cohen. And since this time, we've had a team of people join this group, all on the Simon Fraser side of things, and they're just depicted below here. But most of what I'll be talking about today is the work of Mike Meanwell and Steve Silverman. And so one of the first reactions we looked at then was this alpha fluorination aldol reaction of this pyrazole substituted acetaldehyde. And so this very small table represents months and months of work. So there are many different things tried here, but what we eventually found is that if you treat this aldehyde with proline and NFSI in DMF, and then follow this by the addition of the dioxanone in this case in particular in acetonitrile, we could get fairly good yields of these fluorohydrins as essentially a one-to-one mixture at this fluoromethine center, and with reasonably good enantiomeric excess of both diastereomers. So this was quite exciting, and we looked at the reduction of these compounds, and that proceeded smoothly. And these materials are actually quite stable and isolable and can be stored for quite some time. And after reduction, we then examined the cyclization of these things. And after a large screen of conditions, we found that we could cyclize these molecules either using a fluorophilic Lewis acid, such as scandium triflate, or simply with base. And in the case of scandium triflate, with this pyrazole substituted compound, we found that essentially just one of these epimers cyclized, and we recovered the beta anomer selectively in about 40% yield from a mixture of these epimeric fluorohydrins. With sodium hydroxide, we saw both of these compounds could cyclize, and we got a one-to-one mixture of the alpha and beta anomer. And if you let this reaction proceed for longer amounts of time and use a larger excess of sodium hydroxide, we recovered only the beta anomer in quite good yield, showing that this alpha anomer is being converted to the beta anomer in situ. And so we were interested in whether this process actually proceeds via an SN1 or SN2 pathway. And so we're able to separate these two fluorohydrins. So the syn fluorohydrin and anti-fluorohydrin structurally characterize these molecules and show that if we treat the syn fluorohydrin with base, we recover exclusively this beta anomer in really good yield. And likewise, if we react the anti-fluorohydrin with base, we recover the alpha anomer. But if we let this reaction go for longer, this is then converted to the beta anomer, showing that the cyclization, at least in the case of pyrazole, proceeds via an SN2 mechanism. So we reasoned at this point that we had a real platform now for making some unusual nucleoside analogs. And so the idea then would be to generate these alpha heteroaryl acetaldehyde derivatives, engage them in this alpha fluorination aldol reaction, or alpha FAR as we call it, reduce these molecules to make a 1,3-syn diol, and then cyclize the products of this using either base or Lewis acid conditions. And we refer to this as a annulative fluoride displacement reaction to form this the ribose core of the nucleoside analogs. And I won't show all of the examples, but I'll just highlight a few of these. So certainly in the case of uridine and uridine analogs, this process proceeds really well. And so here you can see the fluorination aldol reaction is essentially quantitative, and the cyclization as well proceeds in excellent yield. 5-methyluridine could be produced. Again, good yields for both of these reactions. The iodo-uridine also was compatible with this process. An interesting example was this C-linked nucleoside analog here, which came from this unusual aldehyde shown on the bottom right here. And you can see that in this case, we have a second cyclization event where the C2' alcohol, the nucleoside, displaces one of the chlorides from this pyrimidine, and this is then trapped in the alpha as the alpha anomer. One of the fantastic things working with Merck is that we could demonstrate some of this chemistry on scale. And this is in the case of this 5-methyluridine, the aldol reaction that was required to prepare this has been carried out on well more than 100 gram scale. And this is just a picture of a bottle of this nice white powder of this fluorohydrin that is, we can keep around for quite some time without any degradation. So some of these aldol products, or these diols, actually don't cyclize with base, but have to be cyclized with Lewis acid. And here's just 3 examples here. So taking a triazole, this diazoadenine, or this phthalimide, all of these had to be cyclized with Lewis acid, either scandium triflate or indium chloride. But again, we can prepare these correctly configured nucleoside analogs now in quite good yields. And again, in these 2 cases, at least demonstrate this on multi-gram scale. Some important things that come out of this process are that these C5' and C3' alcohols are actually protected because these are the original alcohols in this in the dioxanone that are protected as this acetonide. So immediately we've solved one of these challenges that can occur in if you want to functionalize the C2' position of these molecules, is we've already orthogonally protected these nucleoside analogs. Also, if you're interested in making enantiomeric nucleosides, these L-configured nucleosides or nucleoside analogs were readily available simply by swapping out the proline from L-proline to D-proline. So we reason now that this aldol product would be a really versatile building block for nucleoside analogs. And I'll just go back to this example of the C4 modified molecule MK8591. And we were keen to see that if we added Grignard reagents to the carbonyl, could these molecules still be cyclized to make C4 modified nucleoside analogs? In this case, it certainly works quite well. So just taking this aldol product, which is available in 2 steps from commercial materials, we can add methyl magnesium chloride. We generate this tertiary alkoxide intermediate. You just warm this to room temperature and it cyclizes in the same reaction flask to give us this C4 modified nucleoside analog now in just 3 steps total. And we were keen to look at the scope of this. And so we examined a variety of nucleobases, a variety of Grignard reagents, and found that these were generally compatible with this one-step Grignard addition cyclization process. And I just show some of the yields here. One thing I do want to highlight, though, is that the stereoisomer that we recovered from these reactions was generally incorrectly configured at C4'. So this is not the natural configuration at C4', and I'll come back to that a little bit later in the talk. I think we have a good solution to this now. So having access to C4 modified nucleoside analogs suggested that we might also be able to make locked nucleic acids. And these are molecules that have a bridging methylene between the C4' position and this C2' alcohol. And the reason for this is that it locks these molecules in a RNA like C3' endoconformer, or this north conformer. What's been found is that this locking group increases the stability and specificity of these molecules, and so they've become really important in antisense oligonucleotides as building blocks. That said, they're often very difficult to make. And one of the really nice examples that came out of our Grignard work, I would say was unexpected, but really a beautiful example of an LNA synthesis. So if we take this aldol product and we add alkynyl magnesium bromide and dichloromethane, we got about a 40% yield of this tertiary alcohol now with this alkyne connected. And if you simply take the same molecule, and we were just trying to cyclize this to make the C4 modified nucleoside analog, we actually recovered this LNA. So this affects both the cyclization to make the ribose core, and because the alkyne is situated so closely to the C2' alcohol, we get a second cyclization event that gives us this bridging, the bridging group, and forms this really unusual locked nucleoside analog. And I'll just point out that this molecule is made in just 4 steps. And the only comparable molecule is the uracil analog, which required a 23-step total synthesis. And so I think this highlights some of the versatility of these types of aldol products for making nucleoside analogs. Some other things that we can do with these readily available aldol products, you can carry out a reductive amination and then treat with base. And here we can form an amino nucleoside analog now with a nitrogen in the ring rather than an oxygen. We wanted to show that this chemistry was also compatible with the synthesis of deoxynucleoside analogs. And so we combined both the C4 modifications, so adding a Grignard reagent to the carbonyl and cyclizing with a deoxygenation process. In this case, just a standard radical deoxygenation to give this deoxynucleoside analog that's modified at C4. One last thing that we wanted to demonstrate early on was how scalable these processes were. And we were interested in intercepting an intermediate in the synthesis of uprophosphivir. in particular. And so starting with this, with uracil, about 0.7 kilograms of uracil, Merck was able to demonstrate that we could generate this aldol product in about 400 grams as a 1-to-1 mixture, again at this fluoromethine center. And of course, that doesn't matter because after reduction and cyclization, both of these compounds are converted to a single orthogonally protected nucleoside analog here, or nucleoside core of uridine. And this could then be converted to an intermediate in the uprophosphivir synthesis by oxidation of this unprotected C2' alcohol and deprotection in situ to make to reveal the diol function, followed by addition of methyl magnesium bromide to make the tertiary alcohol at C2' here. So at this point, I think we were quite pleased with the versatility of these aldol products for accessing a variety of nucleoside analogs, both on scale and in, I'd say, in many cases in quite general ways. Some challenges though that we identified, and I've already mentioned this, most or almost all of our C4' modified nucleoside analogs had the wrong configuration at C4' if you're targeting natural configurations for these types of things. With regards to locked nucleoside analogs, we demonstrated we could make one, but this wasn't really by design. And so we really wanted to find a more general way to make these types of molecules. And up in the top right here, I show an example of a bicyclic nucleoside analog. And clearly this would not be accessible using the chemistry that we had developed. So I just want to spend the last few minutes here highlighting how we've recently tackled some of these problems, and I think have some good solutions to accessing these kinds of molecules. And I'll just point out that these bicyclic nucleoside analogs are important both for oligonucleotides and in drug discovery efforts. And on the left here, there's a molecule, this bicyclo DNA analog that's been prepared by Lohmann, and who studied a variety of these bicyclo DNA scaffolds for oligonucleotide synthesis. And this molecule on the right is a bicyclic nucleoside analog that was reported just last year by Merck as a PRMT5 inhibitor. Now, these molecules often take many steps to assemble, and so we thought that we would be able to tackle this problem using the chemistry we had established. Now, obviously, as I mentioned, if dioxanone is the aldol coupling partner, we're left with an oxygen at C5 and at C3, and there's no chance now to put this second ring in here. And so to get the second ring in, we would need to have a different ketone coupling partner. So we carried out a screen of a variety of ketones, looking for things that are compatible with this process. And these are just some examples on the top of the slide of ketones that we looked at. And what we found is that these four on the left work actually quite well. So thiopyranone, cyclohexanone, this oxidized and protected acetone derivative, or this pyranone, the bis-TBS ether here, as well as cyclopentanone, work, but not well, and the other ones just did not work at all. And there are several others that didn't work in this process. But what this means now is that we can use these types of ketones to access these bicyclic nucleoside analogs. And I'll just show just a few examples of this. So on the bottom left here, we have examples where cyclohexanone was engaged in a fluorination aldol reaction with this phthalimide substituted acetaldehyde. Or we could take the same aldehyde and use thiopyranone, and you can see the yields for the aldol, the diastereoselectivity, and the EEs are all quite good. Now, these molecules can be reduced just like we'd done previously and tricked into cyclizing using a fluorophilic Lewis acid, in this case, indium chloride in acetonitrile, and we get quite good yields over the sequence of reactions now to prepare these bicyclic nucleoside analogs. Now, a really nice example, and this was actually carried out by Steve Silverman at Merck. He showed that we could take this diazoadenine substituted acetaldehyde, carry out a fluorination aldol reaction to prepare this mixture now of two epimeric aldol addicts. So a 3-to-2 mixture, slightly favoring the syn-fluorohydrin over the anti-fluorohydrin. In this case, these molecules were separable, so these were separated and cyclized separately, using the same conditions. So just the 1,3 syn selective reduction of this carbonyl, followed by treatment with indium chloride to make these two bicyclic nucleoside analogs now with a diazoadenine attached. And you can see that these compounds are made in just 4 steps from commercial materials, And they start to closely resemble molecules like this PRMT5 inhibitor from Merck that take many steps to prepare. And so we're quite keen now on extending this chemistry to the preparation of molecules like this. So the last two things I just want to briefly talk about is some of our work that's focused on improving the diastereoselectivity of these C4 prime modifications, and has now allowed us to maybe more properly access or target locked nucleoside analogs. So if I go back to the original example, so this is the example I showed several slides ago, where we took methyl magnesium chloride and THF. We generated an anti-diol in this case, which cyclized in situ to form this C4 prime modified nucleoside analog. And this was a really fantastic result. I think the fact that we can make something like this in 3 steps was really inspiring to us. But we needed to go back now and see if we couldn't adjust the stereochemistry at C4 prime. And I think anyone that's worked on systems this complex, or sorry, maybe I'll just point out that this stereochemistry at C4 prime is obviously established in the 1,2 addition of this Grignard reagent to the carbonyl. And so working in a system this complicated, there's many variables. So we obviously have a free alcohol. We have many sites for coordination to a Grignard reagent. We have this nucleobase. We could obviously play with solvent and the Grignard reagent itself. And so we decided to simplify things here and start with just an aliphatic fluorohydrin. So this, rather than having a nucleobase attached to this, we just have this isopropyl group. And first of all, examine the effect of solvent and Grignard reagent. And I won't go through all the results here. I'll just summarize, I think, what has been some of the most interesting things that we found. First of all, we found that solvent plays a big role in this reaction, which I think is not surprising, especially considering the obvious chelation that's involved. In particular, we saw up to 6-fold increase in diastereoselectivity, favoring now the syn-diol if we switch from THF to dichloromethane. Something else that we found that's quite interesting, I think, and was really unexpected, certainly by me, was the effect of the halide on magnesium. And we saw a 3.5 or roughly 3.5-fold increase in diastereoselectivity, simply switching from methyl magnesium chloride to methyl magnesium iodide. One other thing to point out is that if we protect this alcohol as a TBS ether, we have complete selectivity for the anti or undesired diol. Again, clearly showing the importance of chelation in this reaction. So we wanted to make sure that these results translated to a system that incorporates a nucleobase. So we went to this thymine system, and again showed that using dichloromethane as a solvent, going from methyl magnesium chloride to bromide to iodide, we saw a similar increase in diastereoselectivity, now 6-to-1 in the optimal case, favoring the desired syn-diol now. Also, you'll see here, if we use dimethyl magnesium, we get essentially a 1-to-1 mixture of diastereomers. And so this gave us some insight to what was really going on here. And so some things that we've learned are that certainly the nucleobase itself has almost no influence on the diastereoselectivity of this reaction. The fluoride also has no real influence on the diastereoselectivity. We can do the chemistry, at least in the aliphatic sense, with a chlorine here and have the same results as we get with a fluorine. The free hydroxyl group is necessary for syn selectivity. And I don't have time to talk about this, but we've carried out DFT calculations that suggest that all alkyl magnesium halides or methyl magnesium halides should be syn selective. One last piece of evidence for some that gave us some insight into this was the lack of selectivity with dimethyl magnesium, which really suggests a key role for a Schlenk equilibrium in this reaction. So essentially what we feel is going on is we have deprotonation of our alcohol using our alkyl magnesium halide. We generate a coordinated species that can then engage in a reaction with a second equivalent of alkyl magnesium halide, which proceeds via this chelate, and 1,2 addition favoring this syn-diol, which is consistent with our DFT calculations. Now, the other thing that can happen, though, is this alkyl magnesium halide can undergo ligand exchange with magnesium, generate this alkyl magnesium alkoxide intermediate, where the 1,2 addition now comes from the undesired face, giving the anti-diol. And so using this information, we've been able to go back now and look at some of these original examples of Grignard additions to these aldol products. And now using the alkyl magnesium iodide and dichloromethane at -78°C, we can favor production of this desired 1,3 syn-diol and affect cyclization to the correctly configured now C4 modified nucleoside analogs. And these are just 4 examples of molecules we've made recently. These yields are simply for the cyclization, showing that the stereochemistry at C4' really doesn't affect the yield of cyclization all that much. These are all similar to the yields for cyclization if we had had the anti-diol. And so now we can have methyl, ethyl, cyclopropyl substitution at C4, and again with the correct configuration. And we've also done this with nucleobases such as this diazoadenine. In this case, we need to cyclize with the Lewis acid, but again, in quite good overall yield. So returning to this challenge of LNA synthesis then, armed with some knowledge about the addition of these organometallics to these aldol products, we went back and looked at the addition of the Grignard reagent derived from chloroiodomethane, the reaction of this with isopropyl magnesium chloride. And now, unfortunately, in this case, for stability reasons, we needed to carry out this reaction THF and MTBE, but we can get about a 70% yield of a mixture of epimeric chlorohydrins now. And this is, incredibly densely functionalized molecule. But if we treat this with base, what we found is we first get a cyclization to form what would be the locking ring in an LNA. And in the same pot, we get a second cyclization to form the ribose core. And I think even though the yield is just 40% for this is a 4-step synthesis now of a protected LNA, which I think is really fantastic. Another nice example of this chemistry is shown here. So if we take dichloromethane and butylithium now at minus 110 degrees Celsius, we can generate the dichloromethyl anion, add this into the same ketone, and generate this adduct now. And again, quite good yield, but as a 1-to-1 mixture of diastereomers. And again, unfortunately, we're a little bit limited here, both by the temperature and solvent with regards to the stability of the dichloromethyl lithium species. This molecule can also be cyclized. So now we can, again, treat this with sodium hydroxide. The first equivalent of base displaces a chlorine from this dichloromethyl group to form the locking ring now with a functionalized bridge. And the second equivalent of base then affects the cyclization to form the ribose ring. You'll see here, I show this as this hemiacetal. We can actually isolate the chloro species, but the yields are a lot lower, and we tend to just derive this to full conversion to the hemiacetal, because that's always a byproduct when we're trying to isolate the chloro product. But again, this can be made now in quite good yield in just 4 steps from commercial materials. So I hope I've demonstrated that these aldol products are, first of all, very easy to make in just 2 steps. They're stable, scalable, and quite versatile as building blocks now for making this wide range of nucleoside analogs. And we're really quite excited to continue to work with Merck and other partners now in producing these types of molecules and supporting medicinal chemistry efforts. It remains, again, just to acknowledge the students that have worked on this project. So the ones in blue are ones that are currently working on the project. In red are all people that have left the lab but started the project, in particular Mike Meanwell, who made this the focus of his PhD work. And of course, our collaborators at Merck, Elsie, Steve, and Ryan, who were really integral in developing this chemistry. And just for those of you who don't know where Simon Fraser is, this is just a picture of it here on top of Burnaby Mountain. You can't see it in this picture, but Vancouver is just about 20 kilometers west of the campus here. And again, I just want to thank the organizers for the opportunity to speak today. And I'd be more than happy to try and answer questions if anyone has any.
Does anybody have any questions? So I don't see any open ones. I'll ask, so has anyone, so, you showed that interesting result with the dichloro pyrimidine kind of at the beginning. Has anybody on the team tried to open that to make a C2 substituted pyrimidine? You could I guess imagine making a uracil derivative with the unnatural configuration.
Sorry, Steve. Which compound were you talking about?
After the alpha fluorination aldol, at the beginning of the talk, you know, something you had showed was that the alcohol actually reacts with one of the chlorines on the pyrimidine ring.
Oh, yes, of course.
Yeah.
Um,
I don't know if anybody's actually looked back at that and maybe tried to open it.
Yeah, no, we just left that result. No, we did not look at opening that, but I think you could open this and probably get a C2 inverted, well, a C2 inverted nucleoside analog by opening this. I think it's primed for that kind of chemistry.
Yeah, that could be an interesting compound.
Hey, Rob, I have a question here. So it's not often you see sp3 fluorines like that being such good electrophiles. I mean, obviously you've got you're making an intermolecular bond, but does the fact that it's got an alpha heterocycle, N-linked alpha heterocycle, make it more labile to these reactions?
Yeah, 100%. So this is, I mean, we look at this a lot like a glycosyl fluoride, which are, you know, well-established donors in carbohydrate chemistry. I think without the N-linked or in the case here, the C-linked heterocycle, these help in the process of displacing the fluoride. We've done some work totally unrelated to this on this displacement of benzylic fluorides using Bronsted acids to promote that process. So I mean, you know, I think we need to think about fluorides maybe as not as inert, and I think medicinal chemists would probably know this, that they're maybe not as inert as we like to think, but certainly in these cases, this whole process is enabled and is facile really because of this N-linked heterocycle.
Yeah, it's great chemistry. Nice job.
Thanks.
So there is one question. So we'll just, I guess we're just about out of time, but we can go through this quickly. Nick Shapini says, great talk. Can you funnel the dichloromethyl addition product to a ribonolactone nucleoside?
Okay, so this is at the very end. So this dichloro addition product. So this is something we've only done in the last few weeks. So we've first of all just been trying to figure out, I mean, to start with, we've been trying to figure out whether we could generate the chloro product in good yield and then use that as a building block for making various bridged LNA analogs. But yeah, certainly I think we could oxidize this to a lactone. And it's one of the things that we will be looking at.
All right. Well, we're right at time, so I think we can stop here. So thanks to both of the speakers, and also thanks to the CCHF for giving us this platform, Division for Organic Chemistry. And yeah, I think we can stop it right here.
Thanks, Rio. Thanks all the speakers, and thanks all the attendees.
Thanks.
Great job, everybody.
Yeah, great job. Sorry I couldn't join you at the start.